Hi, I'm Chandra Almond, and I'm here to talk about the SC2I. And the title of my talk is Beyond Clinical, Integrating Research Assay Data into the Observational Health Data Sciences and Informatics Common Data Model, or OHDSI CDM, through the SC2I, or the Surgical Critical Care Initiative. So SC2I is funded by the DoD, or the Department of Defense, and it is a consortium of institutions which aim to improve the care of critically ill and traumatically injured individuals by facilitating the development and implementation of CDSTs, or clinical decision support tools. SC2I is primarily focused on traumatically injured adults. Because it's funded by the Department of Defense, it is primarily focused on military wounded individuals, but obviously we also enroll civilian traumatically injured individuals, and other patient controls include critically ill patients, scheduled or non-traumatic surgical patients. So we do have patients that are septic in nature, or patients with a scheduled coronary artery bypass or a CABG surgery, or things of that nature. Our prominent research questions focus on how we can take the data, both clinical and biospecimen or assay data, and use that to predict clinical decisions, right? So some of the studies that we've done, or some of our particular research questions, are focused around severe traumatic brain injuries, or STBI, time to close open wounds, whether that be extremity or abdominal, as well as algorithms to determine the predictive need for MTP, or massive transfusion protocol. The primary and largest protocol, which is going to be the focus for today's talk, is the Tissue and Data Acquisition Protocol, or TDAP. TDAP includes about 12,000 data points at each time, over 30 time points. TDAP includes approximately 1,100 patients, primarily trauma, and the protocol collects blood and urine on every patient, as well as potentially cerebrospinal fluid, CSF, wound effluent, peritoneal fluid, etc. Basically, TDAP can get any exempt biospecimen. That it means it's not going to be needed for clinical diagnostics at that time. The SC2I is interested in aligning clinical data with the biologically derived data to assist in CDST development. And the clinical data set is quite large. It's about 12,000 variables, and that includes ICD codes, CPT codes, so basically about surgeries and diagnoses, medication use, and course of care or treatment. So we might have length of stay or ICU days if somebody was on a ventilator, things like that. The assay results is just as large, or almost as large. We collect cytokine and chemokine, flow cytometry, qPCR, microRNA, mitochondrial DNA, protease activation. There's a lot that we capture. These are the primary ones of interest that we tend to use on most of our research questions. So the TDAP data set is about 1,100 patients. There's actually more than that, but that's what's in the database. We are, I think, going to grow to about 5,000 over time. And of those patients, 72% are traumatically injured. We have about 8,000 time points, and about 90, that says 90,000, it should be 90 million data points across the time. So before transitioning to OMOP, we had one large flat file. So all those 12,000 variables by those 8,000 time points. So it could get pretty cumbersome to try and make sense of So we had ultimately just determined we were already going to do a relational model. In 2020, we started the transition to the OMOP CDM in hopes that the standardized vocabularies would be perfect match for our translational database. So we started mapping, and we noticed that the majority of our research assays, unfortunately, were lacking in the OMOP CDM. So what we found was that a lot of groups had focused research questions, whereas our research questions tended to be much more exploratory and model development. So we might have 100 variables and we see how they are linked, and then we come up with an algorithm from there, as opposed to saying we have two, maybe we have IL-2 and IL-6 we think are going to be high in this group. Instead, we're looking much broader. So we look at a full panel. So of those 12,000 assay variables that we have, only about 200 were available in the OMOP already. So we created new mappings in the measurement table and included rows that kind of extended out into the ancestor concept and the concept of relationship table. And so what that means is the addition of those SC2I assay concept code and relationship hierarchy were the expansion will mean that future researchers will be able to use those vocabularies and be able to essentially enable a translational model in the future. By adding these research assays to a predominantly clinical data model, the hope is that we are creating more of a translational model for the future, so that others can create biology-driven CDSTs and implement new precision medicine research. So we are very excited for what is to come, and we are in the process of submitting those to OHDSI, and we anticipate that they will be accepted as standard models. And I'm happy to share anything, any particular mappings or results with others as you're interested. Thanks for listening.